Good morning, everybody. Today's going to be short but dense. We're going to talk about alternate splicing, which is a way to get more than one different protein from the same gene or same primary RNA transcript. We use this mechanism a lot as mammals. We have only about 25,000 protein coding genes approximately, but the fact that each one can be used in multiple ways to produce different proteins explains how diverse our proteome is compared to the number of genes we actually have. This happens really quite a lot in mammalian RNAs. Okay, we're going to do a couple of classic examples today. One is a calcitonin and CGRP gene in human, which is spliced to form two different peptide hormones that are active in different cells. And then two Drosophila proteins, sex lethal and TRA, which govern sexual development in flies. Okay, so there are a lot of different patterns of splicing that can occur, and these illustrate some of them. Basically, anything that you can think of might happen probably does somewhere. In this example, we have the mutually exclusive exons, for example, where we have a pink one and a blue one, and every primary RNA transcript is spliced such that either the pink one or the blue one is included, but not both. Sometimes you have cases where introns are actually retained. They're not spliced out in at least some versions of the primary RNA. You can also have cases where there's different 5' and 3' splice sites that are used. For example, we have the pink section of an exon where sometimes the 5' splice site is actually within the exon and sometimes it's at the other end, with the result that you will have slightly different proteins with those last few amino acids coded by the first exon, etc. So there's many different ways that you can splice or not splice to give a great diversity in your proteome. Okay, so our first example is going to be calcitonin, which is a thyroid hormone contributing to calcium regulation. Okay, so this is actually produced ultimately by the same gene that produces the CGRP, also a hormone, this time instead of in thyroid, in neuronal cells. So calcitonin splicing is regulated by a calcitonin-specific splice acceptor, which is activated only in thyroid cells. And you'll see a little bit more about that in a sec. Okay, so here's a diagram of the gene which encodes both of these peptides. We have exons 1, 2, and 3, which are present in all versions of the final mature RNA. But the calcitonin is actually encoded by exon 4, which is not included in the final RNA in neurons, but is included in the thyroid. Alternatively, we have the CGRP peptide hormone exon, which is included in neurons and not in thyroid cells. So the way that works is that exon 4, the calcitonin one, is flanked by weak splice sites. So it's not a good signal, or it doesn't provide the proper signals really for the splicing apparatus to see it very regularly. So what is included in an intron adjacent to it is an intronic enhancer element. So what that does, of course, is to bind proteins which will facilitate recognition and usage of the adjacent splice sites. So this intronic splicing enhancer adjacent to the exon is active exclusively in thyroid cells, which is why the calcitonin exon is included in those cells, which is not, CGRP is produced only in neuronal cells. So what they do, CGRP is not really well understood, but it does function in pain-related pathways, such as wound healing. Okay, so it's important to recognize that it's produced by the same gene that produces calcitonin, but the roles of those two peptides are completely different in cells. Here's a final picture of what the splicing looks like. The calcitonin gene includes several exons, one of which encodes calcitonin, and the other of which encodes CGRP. And splicing goes differently in thyroid versus neuronal cells, such that the calcitonin coding exon is included in the final RNA in thyroid, but not in neuronal cells. And conversely, the CGRP peptide coding region is included in neurons. Now, there's also alteration of the poly A sites, which we don't need to worry about right now. So those primary transcripts, of course, are then going to be translated and are actually processed. The final peptide hormone is processed out of the translation product to finally produce the active peptide, which have no sequence similarities to one another at all. Okay, so welcome to our favorite model organism, Drosophila melanogaster, which is a fruit fly. We're going to be talking about sexual development, at least some parts of the sexual development in those flies that take place in the early embryo. Okay, so Drosophila is a very important model organism. For classical genetics, a very early definition of genes and chromosomes, in fact, and understanding of biochemical pathways, etc. But it's still very popular and very important for things like developmental biology because of the high degree of conservation of developmental genes across metazoans. Very popular for neurobiology, especially because the way neurons work and develop is a very conserved process and can be very informative of what happens in other organisms. Also popular in aging, evolutionary biology, behavior, etc. So what we're going to talk about is sex determination in Drosophila, which superficially looks like what we do in mammals, but the mechanisms involved are totally different, right? Flies can be males or females, XX or XY. So can we. But it's very different from ours because if you're a mammalian female, you've got two X's, and you're a mammalian male, you have only one X. So you have to have a way to alter the dosage of one or the other in order to equalize the dosage between the sexes. Now, of course, in mammals, what we do is we inactivate one of the X's in all female cells. In Drosophila, it's totally different. What they do is increase the expression of the single X chromosome in males. So even at the level of the dosage compensation system, the two systems are really totally different from one another. Okay, so just an overview of what this is going to be about, where we're kind of going with this. We're going to be talking about two genes, really. One is called sex lethal, that plays a role in sex determination. And sex lethal is a splicing repressor, and its job is to guarantee that female embryos will develop as females. It's part of a complicated cascade of proteins. And the females will produce this sex lethal protein, and male ones do not produce it for reasons that have to do with the way the initial primary RNA transcript is spliced. So here's an overview of what's going to happen. We've got XX females. Now there's a very complicated system of events that occurs in the very, very early embryo that ensures that the XX future female embryos have a little bit of sex lethal protein to get the process going. XY male embryos do not produce that early burst of sex lethal, and so they don't ever actually activate that gene or the female-specific splicing pattern, as we will see. Okay, so here's a slightly older diagram, but I think it illustrates what we need pretty well. It does not include all the exons, just the ones under consideration for the splicing changes that occur in future females and future males. Okay, so the top part has sex lethal itself, which actually autoregulates its own splicing. And we're only showing exons 2, 3, and 4 in this case. So sex lethal is a splicing repressor, and it binds adjacent to the third exon, and that's the one we want to exclude. Females, or the active form of sex lethal, includes exons 2 and 4, but skips exon 3. Male embryos include sex lethal exons 2, 3, and 4, which is to say include 3, the one that is skipped in females. Well, that is important mechanistically because there's a stop codon in frame within the exon 3 that causes the production of a shortened protein in males, which is inactive in terms of the sex lethal activity. So the splicing repressor activity of sex lethal guarantees exon 3 will be skipped in females or future females, whereas it will be included in males. Here's a better picture of what that looks like. We have the 5' splice site at the end of exon 2, ready to go to splice to exon 3. But we've got the little blue dots that are sitting there, which are binding sites for the sex lethal protein. So what happens is in early female embryos, the sex lethal protein is expressed, binds to those repressor sites. So those would be considered intronic splicing repressor sites. and prevent basically the splicing system from recognizing that 3' splice site at the 5' end of exon 3, with the result that exon 3 is skipped and that you get a protein which is active, which includes exons 2 and 4. Now remember, exon 3 is the one that has a premature stop codon, so if you include it, you end up with an inactive protein that can't govern any other activities that sex-lethal normally would do. Okay, so this is what I just said. The sex-lethal binds to 2 sites that flank the exon, inhibits the use of the 3' splice, and the effect is that exon skipping, and as a result of this particular situation, the production of sex-lethal leads to more production of itself, because we're talking about the splicing of its very own transcript. Okay, summarizing what I just said, if exon 3 is included in the mRNA, you will produce a non-functional protein. So males do that because they don't have a mechanism in place to skip exon 3, which has the stop codon in it. So the male form will have exons 1, 2, 3, and 4, but the fourth exon is not translated because of the stop codon at exon 3. In females, we exclude exon 3, and that gives us an active final mRNA, which contains all the exons except 3, and that produces the active sex-lethal protein. Okay, so this is summarizing what I just said. In males, exon 2 is included, and that contains a stop codon, which you don't want because it prevents full-length TRA from being produced. So active TRA is produced only in females, and that allows the next step in the cascade, which is to say the differential splicing of the double-sex transcript in males versus females to produce 2 different forms of this transcription factor. So here's another picture. Here's a picture of TRA again. Okay, so the sex-lethal binds adjacent to that exon. It makes it skip that one splice site and then produces a TRA protein, which does not have the stop codon and allows active protein to be made. So the action on double sex is kind of the opposite of the action on the other two, sex-lethal and transformer, because the TRA is actually going to facilitate splicing. So in facilitating splicing, there's a weak splice site at the 3' and 3' splice site in exon 4 is weak and needs help. So the binding of TRA will facilitate the use of that upstream splice site in order to allow exon 4 in its entirety to be included in the final RNA. So in this case, the job of TRA is not to inhibit splicing, but rather to facilitate it by allowing the use of a weak splice site at that exon. So the ultimate effect of that is that females will include exons 3 and 4, and males will skip 4 and will include exons 3 and 5. So this produces 2 different forms of the double-sex protein ultimately, one double-sex female and the other double-sex male, and both of those are active in terms of guiding further sexual development in these flies. Okay, so DSXF is a double-sex female, DSXM is double-sex male, and remember, TRA is present in females, but not males, and that's how we can produce the double-sex female form of this transcription factor. All right, so TRA facilitates the use of a 3' splice acceptor in double-sex, allows exon 4 to be included, etc. So you want to have exon 4 to be skipped in males and included in females to get the 2 different forms of this transcription factor. Okay, so now we've got a few practice questions. This one's not hard. We have a Wilms tumor protein, WT1, and this is true. There's actually 4 different forms of this protein, 4 different isoforms, and they are determined by how the splicing goes. Okay, so there's 4 forms, and this is actually pretty simple if you think about it, right? We have 2 regions which may or may not be there in the final protein, and the reason they are or are not there in the final protein is whether or not the exon that included them was retained or skipped during splicing. So the exons under consideration is exon 5, encodes that 17 amino acid piece, which may or may not be there in a particular isoform, and then the KTS region, which is encoded by exon 8. So those are the alternative exons that may or may not be included in any final RNA, depending on how the splicing goes. So the plus 17 plus KTS, those would be resulting from transcripts in which both of those exons were included. Neither one was skipped. The minus 17 plus KTS come from RNAs that were spliced such that exon 5 was skipped, but exon 8 was included. Plus 17 minus KTS are ones that included exon 5 and skipped exon 8. And finally, we have the ones that are minus for both of those, and for those came from RNAs where exons 5 and 8 were both skipped. So it looks like a fairly intimidating question, but it's really actually pretty simple. Okay, so one mechanism is what I just said, that there's the calcitonin has a weak splice site, well, the 3' splice site between exons 3 and 4 is weak, and that it needs help, and that you need to have an intronic, or exonic, I suppose, splicing enhancer to bind specific proteins that will then facilitate recognition and use of that 3' splice site. Well, that's the actual answer, but there's more than one other possibility. I mean, another possibility would be the calcitonin itself is normally suppressed, and by a intronic or exonic splicing repressor sequencer, splicing silencing sequence, and that needs to be overcome in order for splicing to occur. So just seeing what happens doesn't tell you how it happened necessarily, and there's often at least several different ways in which you can explain observations, which of course is why you have to do experiments. So anything that makes sense is correct in this setting.